Plasmid stability. It is another factor that is important in controlling the expression of a particular gene in the host organism. अगर हम ये सपोज करें कि हमने जो एक्सप्रेशन है किसी ख़ास जीन का उसको मैक्सिमाइज कर लिया है, तो हमें ये भी जहन में रखना पड़ेगा कि इस मैक्सिमाइज एक्सप्रेशन का या ज़्यादा एक्सप्रेशन का प्लाज्मिड पे क्या इम्पैक्ट होगा जो कि सेल के अंदर है. तो दिस फैक्टर it is also very important, because if the expression of the target gene is very high, then it may leads to the decrease rate of cell growth. If the cell growth rate is decreased, then another factor that may be there, that morphology of the cells, it may be altered. Alteration may includes that the cell may show increased filamentous growth, and the cell also will exhibit increased fragility. So these are the impacts of maximize expression of the target gene. So if expression is high, so it can alter the cellular structure and cell fragility. So if expression is high, so we can say that it can also lead to other impacts, that cell may lose the plasmid altogether, or some structural instability it is there, and even the plasmid, which is recombinant plasmid, it may be present in low copy number. So all such problems may be there when we will try to manage the high expression of the target gene. So plasmid stability we can discuss in terms of segregative instability and structural instability. If we focus on segregative instability, I think we also focused somewhere in the previous lectures that segregative instability, it is the ability of the plasmid to segregate or to maintain equally after cell division. When cell division is there, genomic DNA as well as plasmid DNA, it is distributed equally. And especially in case of natural plasmid, it is managed, because in natural plasmid there is a region of the DNA in the plasmid which is called as PAR region, PAR from partition. It can manage the equal distribution of the plasmid into the daughter cells. On the other hand, artificially constructed plasmids, like in case of PBR322, initially this region was deleted, because this plasmid, which is derivative of cold-even type of natural plasmid, it is present or maintained as high copy number plasmids. अगर इसका कॉपी नंबर हाई है तो सम टाइम क्या होता है कि पार रीजन के ना होने की वजह से अनइक्वल डिस्ट्रीब्यूशन होती है कि एक डॉटर सेल्स में प्लाज्मिड ज़्यादा चले जाएंगे, दूसरे में कम चले जाएंगे. So it is not so much problematic, because if unequal distribution is there, at least daughter cells they will contain certain level of plasmid. Some may be high, some may be low. But it become problematic if one daughter cell it contain all the segregated plasmids, another lack the plasmids. But it is very rare event in case where the plasmid they are present in high copy number in case of PBR322. On the other hand, if the PAR region from a natural plasmid like PSC101, it is cloned or inserted into the PBR322, then its equal distribution, it is manageable. So the presence of partition region or PAR region, it is very important to maintain the certain level of plasmid copy number into the daughter cell. But In addition to that, some other factors may also be involved in order to manage the copy number into the daughter cells. For example, certain mutants have been used that are deficient in the PAR region, and here the superhelicity of the DNA, it has contributed to the maintenance of certain plasmid copy number into the daughter cell. So in addition to the PAR region, superhelicity of the DNA or plasmid DNA, it is also important in order to determine the segregative stability of the plasmids into the daughter cells. And I think we have focused on this topic, especially that how superhelicity or certain mutants they can manage the equal distribution of plasmid DNA into the daughter cells.